Afternoon. I hope everybody is well caffeinated and ready for the exciting sessions of Wednesday afternoon. There's an adage about surrounding yourself with people who are accomplished and people who can teach you, and I'm very fortunate to have learned in my 6 months with the foundation that the next moderator is in fact one of those people. It's my pleasure to turn this over to Kim Butts-Pauley and the Technology Breakthroughs panel.
Thanks so much. This is really just such an exciting time to be in this field because there's so much exciting research that encompasses everything from hardware and image guidance and protocols and drugs and biological understanding. So today we have an array of panelists with a variety of different expertises. And so what I'd like to do is start out the session by asking them to tell us a little bit about, first introduce themselves, and then tell us a little bit about what they've been seeing either in the abstracts that was really exciting or kind of the field in general in their area. So I'm going to just call on them and start with Cyril.
Thank you, Kim, and thank you for attending this session, and thanks to the FIRST Foundation for organizing this. So yes, I've been reviewing a little bit the abstract, the people who contributed to this conference, and regarding, well, you know, in my lab, well, I forgot to introduce myself, sorry. I'm Cyril Lafont, I'm the head of Labto in France, which is part of the NIH, and we have been working for the last 40 years building some device for therapeutic ultrasound. Our main realization is a Focal One for treating prostate cancer, the device for Cartera, the SonoCloud, and other device like IOP1 for treating glaucoma. So looking at the presentation, I've been always interested in finding innovations like specific device, and I must admit that I really enjoyed the presentation by the colleagues from Georgia Institute of Technology. They proposed the use of CMAT, it's a new technology of transducer, for their device for opening the blood-brain barrier, and they perform very interesting tests where they use the same transducer for both opening the blood-brain barrier and listening the cavitation. So this strategy is interesting in my opinion because it gives some flexibility for the probe. You have a transducer that is much more broader bandwidth, so that's very interesting. You can do both imaging and therapy. I'm also interested because we are doing the same sort of transducer for prostate cancer, so not a mechanical treatment, but more a therapeutic thermal treatment. So that's, I think, the transducer of the future, and I wanted to highlight this presentation in particular.
Yeah, thanks so much. CMUTs are something that we've been seeing for many years now because of all of the advantages of them, for example, the wideband, the capability. Why do we think it's taken so long to kind of see more CMUT development in our field?
Well, asking me? Or?
Yes.
Okay.
Unless anybody in the audience wants to step up. Otherwise, Cyril, you're on the spot.
So I think there were some issue with the robustness of the transducer. Some significant progress have been done on this. And now these transducers are able to generate high enough pressure to open the BBB, but also operate for a long enough time in order to ablate tissues, as we've seen in prostate. So we're getting close to bring this sort of transducer in new devices and new clinical applications.
Great, thank you. Okay, so I'm going to go to Hong next, and after Hong, we're going to go to Tanya. So Hong, you're up.
Okay, my name is Hong Chen from Washington University in St. Louis, and I have been assigned to watch many talks. So I'm going to pick only one, specifically in the field of neuromodulation technologies. So this talk, the abstract ID is BRT24. And then this is a talk given by Jung-Yong Park's team from South Korea. So they developed this very unique device. They call it ultrasonal cover slide. So what they do is they get commercial cover slides, and then they just directly attach PVDF membrane, which is just the ultrasound transducer, directly to the cover slide. Now you can do cell culture experiment by simultaneously doing ultrasound sonication with optical imaging. So that definitely make the cell culture experiment much easier to do. this is the one that I like the most.
Yeah, that's great. You know, one of the things that's going on in the field right now in neuromodulation is just the thought that we really need to understand at multiple different levels what's going on, what's effective in humans, but also really what's going on the ion channels and being able to see at different levels. So that's really exciting to have new equipment for that.
Yeah. It solves a lot of issues because when we do the cell culture experiment, how to couple your ultrasound transducer with the cell culture, whatever media, dish or whatever you use, that's definitely challenging. So this solution may not solve all the problems because it's a PVDF membrane, so the output is limited, but at least the concept of directly integrating ultrasound transducer on the cover slide, I believe that can solve some of the issues.
And a very common problem is, of course, standing waves that get set up there.
So what they did is in order to minimize standing wave, they also put absorber directly capped to the objective, so it absorbs the ultrasound wave and then decrease standing wave.
Right, great, thanks. Okay, now we're going to turn to Tanya, and after Tanya, we'll have Eli. Okay. Tanya, can you?
Great. So, can you hear me?
Yes.
Okay, great. So yeah, I'm Tanya Kuklova, University of Washington. My research is mostly in drug delivery using mechanical effects of ultrasound and cavitation for drug delivery and mechanical ablation or histotripsy. And so I'd like to highlight a couple of talks that I found really interesting. One sort of new conceptual understanding of aspects in drug delivery, which actually came from Hong's group. It was on lymphatics and intranasal delivery of drugs, which I found to be really interesting conceptually, in that the drug in that case propagates not inside the vessel, but along the perivascular space and that's done through cavitation inside the vessel, and that cavitation acts like a pump, kind of pumping that drug along the perivascular space. So that was really interesting to see, and I'm wondering, in fact, if so clearly there are applications in the brain and intranasal delivery, but I wonder if that can be used in other parts of the body to transport things along perivascular space as opposed to intravascularly. So that's kind of a question to ponder. And then the other thing I wanted to highlight is somewhat underappreciated technical aspect is acoustic coupling, and an excellent work by Alison Payne's group on a skin cooling pad that is conformal to the body, that they have designed a custom MR-compatible system with a cooling pad that does just that. And not only did they do that, but also put all the technical specifications on GitHub, so anyone could build one for them. So that's something I found interesting lately.
Great, thank you. You know, some of these details are so underappreciated. Maybe we'll just jump around a little bit, because I think I wanted to have a chance to ask Rafi, who had something to say about shaveless. Is one of those details along the lines of coupling. So do you have something to say about that?
Well, yeah, thanks, Kim. So I looked at the title of our session, right? The technological breakthroughs, and shaveless is kind of, there's no technology really, there's no breakthrough, except that we can actually treat patients without shaving them, which is a huge deal for the patients. And when you think about it, shaving and shaving razors are at any ultrasound clinic, right? Shaving is used for diagnostics, it's used everywhere, and we used to shave all the patients, and now recently at Insightec we had the opportunity to gain experience with treating at low frequency, but treating patients without shaving them. And this experience has been great, meaning that essentially today we ignore the hair in the sense that we get the same results with shaving or without shaving.
Mm-hmm. And does it matter what kind of hair it is? Does it have to be really thin hair? Or, yeah.
Yeah, so, I would be hesitant to put Bob Marley inside our helmet and try to open the BBB. So I would think that the amount of hair, the length of the hair, the density of the hair are all relevant parameters. What we try to do for the patients is look at a really worst case scenario, quantify that and see that it's still not an issue. And then we step back a little bit from there, and this is why we do not shave the patients, but we do ask them to take a haircut.
Okay, good. Let's move on to a different topic. So let's see, Eli, and then we'll go to Jen.
Yeah, thank you. So I'm Eli Vlasavlovich from Virginia Tech, and this is a hard question. Obviously, we saw so many great abstracts. So I'm going to focus on a lot of my research is centered around histotripsy, so I'll just maybe mention a couple areas that I've been excited to see in the field over the last 2 years, particularly some of the abstracts here today. And I think those of you who are working in this field know that some of the features we've seen with histotripsy, and now very exciting time as we move into clinical trials for promising clinical results in liver, and some of these features being non-invasive, non-thermal, but really being able to see in real time where we're treating and targeting, as well as the tissue feedback. So now the technical challenges of moving this into different cancer types and different organs. And so it's been exciting to see the different groups that are working on different systems, different strategies to achieve this. And so if we look at just the brain, which obviously there's a lot of interest in this room in targeting the brain with histotripsy, starting with the group at Michigan, we saw, that's my former group, I should put a disclosure in there, but I've been very excited to see how advanced their systems have gotten in the last few years. Particularly, there's a couple abstracts where you can see their acoustic cavitation feedback, so being able to take the real-time nature of our ultrasound imaging of the bubble cloud that we have in the liver and be able to achieve that in the brain, I think is going to be very important. I've also been excited to see that they're working on both MR-guided systems as well as new neural navigation devices, I think. And they have a ton of data. Jen's on this, so I won't talk anymore about those systems. Also, there was an abstract showing using, I think it was the InsightTech system that was modified doing boiling histotripsy in a pig model. Very exciting to see that that's now moving forward as well. And I also wanted to highlight a couple others that weren't in abstracts but have been recently published that are looking at alternative methods using ultrasound guidance. So Dr. Francesco Prada's group has developed an intracranial implant that for treating, and I think this is beyond histotripsy, but being able to do ultrasound-guided real-time procedures in the brain, I think has a lot of beneficial opportunities. I've also been fortunate to work with Jeremy Brown's group in Halifax that's developing miniature handheld histotripsy systems, high frequency for high precision intraoperative ablation in the brain. I think those same devices can be really useful for our different groups when we look at small animal models and testing histotripsy in those models. So overall, I think just seeing everybody working on different strategies and how these different tools are being developed that we can then put in the hands of our clinical colleagues so they have more tools to see histotripsy advance in the brain and similarly as we move into different organs of interest.
Great, let's digress for a moment and talk a little bit about maybe alternatives to MR. So one of them is the passive acoustic mapping. So what's the state of the art there? Do you have comments maybe, Ryan?
Sure. Thanks very much. I'm Ryan Jones. I'm a research physicist at Sunnybrook in Toronto. So that's an area that I've investigated over my time there, and also there's been some great work so far at this meeting on that. So maybe if I can just point out a few abstracts that I thought were interesting. I was going to talk about John Sukovich's ACE monitoring, but you beat me to it, Eli, and I think Jen will also probably talk a bit about that. But I thought it was great that they're able to use these new transmit-receive arrays that they've developed to actually look at the acoustic emissions that they're getting during these violent cavitation events and see what correlates with what in terms of the liquefaction of tissue, for instance. There's also really nice work out of Boston, Nathan McDonald's group, BRT 28 is the abstract, where they looked, I think this is the most probably comprehensive analysis comparing the acoustic emissions that they're able to measure during those brain treatments, in this case in GBM patients, and compare those to the sort of radiographical findings they're seeing, either contrast enhancement or at T2 star-weighted effects. And he saw some nice correlations, particularly within a patient. And maybe the correlations got a little bit muddier when he looked at the population as a whole, which I think he explained very well in terms of various parameters that could be looked at going forward. So I thought that was very interesting work. And then I know I haven't quite got to your topic of mapping yet, but in terms of cavitation-based control, so I think both in Nathan's paper and also I think Dr. Woodworth's talk and Jibo Chang's talk, they were able to show that the new device subspot control from the Intelsec system seems to have really helped increase the homogeneity of at least the cavitation that they're able to deliver, and hopefully that'll lead to more efficient treatments.
Mm-hmm.
So Kim, if I may, it's amazing that we were looking at the same abstracts and thinking more or less about the same things, and I appreciate what you just said. And, I also looked at machine learning and was wondering how does it play in. There's some work coming out of Shanghai Tech, Wenjing Li, if I pronounce her name correctly, also working at the UT Southwestern, trying to use machine learning to predict the BBB opening and tissue effects. It's in a rat model, so it's limited. And also to mention another abstract from Matteo Ginzo, working at Besta with Dr. Prada, measuring the distribution of microbubbles in the brain in a guinea pig perfused brain model, and talking about the same things that Nathan McDonald is mentioning, that there are so many parameters at play, and when you look for these correlations, you don't, especially in clinics, you don't really know what is kept fixed and what is varying. So when I put all of this together, I end up with more questions than answers. But maybe it's a good opportunity to pose these questions to the panel and to the audience. What is machine learning the correct tool in order to predict BBB opening? Is it enough to have a predictive tool, or do we need to actually confirm the BBB permeability or drug delivery when we can still do something about it with a particular patient? One of the things that come out of McDonald's work is that even if you learn something on one patient, with a different patient, you get a different result, and then you need to adjust. And finally, do we really care about all of that, or do we just shoot ultrasound and hope for the best?
So who are we going to get to answer those questions? Does anybody on the panel want to answer, or should we see if Nathan or Costas are available to answer? So I think those are some really good questions as to, do we really need to understand the signal, or just sort of look at the correlation with the result? Yeah. Okay.
Yeah, I know that I cannot answer the questions, but I think it's actually a great sort of this point for me to talk about what I thought is interesting, because I think it's all related to what I also was observing too. So by the way, I'm Zhen Xu. I'm the head of the historypsy group at University of Michigan. And I think all this discussion so far, in terms of technology breakthroughs, and I concur that there are a lot of talks so far on the technology side, and especially from the monitoring and the feedback side, I find it's really interesting. And I just want to take a step back, saying that, for the longest time, right, like in the MRI-guided focus ultrasound, it's a really exciting and a cool technology, and has been allowing us to perform non-invasive brain therapy with safety and good accuracy. But taking a step back, and I was always wondering, the high cost and the long MRI hours required to perform those procedures has been really restricting us from spreading a widespread use of focused ultrasound for the brain treatment, for example. So of course, there have been always studies on neuronavigation guided focused ultrasound, and that's not new. But I think what's new is that there has been this rapidly developing acoustic monitoring coupled with neuronavigation that have been allowing us to actually do treatment outside, potentially outside the MRI scanner for brain treatment. But still with feedback, and now Rafi was posting, and also Ryan, talking about those questions in terms of for BBB opening, for example, the microbubble distribution, right? And there's also cavitation monitoring and all that. We have seen abstracts from the symposium where the PCB coupling with the neuronavigation system for BBB opening from Howley Liu's group, and from for BBB opening, and from Hongchen's group for the sonal biopsy. Also, I think both Eli and Ryan talk about Johnson Sukovich from our group, now using histotripsy transmit and receive capable array that can do cavitation monitoring and mapping and so on. So we do have certainly new technology allow us acoustic technology allow us to do some monitoring for the brain without the requirement of using MRI. The key issue now we identify, right? Like we need to identify certain key issues for different kind of treatment. The microbubble distribution is one, as Nathan McDonald pointed out. Then there are others. The question I have is actually, well, not quite answering, but whether acoustic technology versus MRI technology would do a better job and potentially solving those issues. I do think that trying to directly just look at the results of the correlation and hoping for the best probably is not the best way to go because we do know things like microbubble distribution is a key to actually having good results. So I do think those need to be figured out. But again, the question is what method can we use, whether it's acoustic or MRI, and combined with machine learning can actually solve those issues.
Oh, yes, go ahead.
I think I heard my name, so I don't know if Nathan is still around, so I can just make a couple of comments. Definitely, we should not wish for the best. We should definitely collect data. And I think one aspect that Nathan mentioned is that different brain regions have different vasculature and different echoes potential. And I think CMATS, one of the things that made us move towards CMATS is that this technology is possibly the most sensitive that we have. So this may allow us to capture microbubbles at different brain structures, and especially at the brain structures that we have very low vascularity, therefore low microbubble concentration and low signal. So I think that the hardware there with the CMATS gives us the opportunity to be more sensitive. And the controller aspect, I think for drug delivery especially, we are really now at the early stages. A lot of work is happening in tumors, so if we overtreat a little bit, that will be okay. But if we start thinking about drug delivery and in the context of like having multiple treatments, I think being able to tune the exposure conditions and prevent adverse effects is quite critical. And we have a paper that is coming out and we're presenting that we see that the broadband signal is concentrated at the early few seconds of the administration of the bolus, which provides some strong evidence that whether we have inertial or no emissions, broadband emissions, is related to microbubble concentration. So that kind of like take us to the position that we really need to monitor the acoustic emissions. We really need to have very sensitive technologies. And again, going back to whether we care or not, I think in the long term, we will end up with data sets that we might have questions to answer. And I think having the data and the acoustic emissions might allow us to improve or understand false positives and false negatives. So I think there are many reasons why we really need to record the acoustic emissions, we really need to be sensitive, and we really need to do the most out of this data. Machine learning might play a role there because a simple back-of-the-envelope calculation, if you do one treatment with 64 targets, for example, with the Insight Tech data, you can see how many hundreds of data points you can have. So there it kind of like lends itself, only due to the sheer number of data that we will have, might create more precise machine learning algorithms. But how the machine learning algorithm will be used and what questions they will help us to answer, I think this needs to reflect back to the clinical experience. What are the important questions that we are interested in asking, and how we can use machine learning to potentially reduce the uncertainty or increase our confidence in addressing those questions. So I hope I didn't steal the thunder of the excellent panel, but that's my couple of comments.
That's great. Thank you so much. Any response?
Thank you for resonating with that. I think that machine learning goes hand in hand with the big amounts of data, and yes, I am probably the one to appreciate how much data do we have out of the exablate treatments. I'm not a machine learning expert, but I think what we're missing is a clearer understanding of what we're after and which methods do we want to use to get where we want to go. And there is a need to do it across patients. There's a need to consider, even in the same patient, the bubble administration method and quantity. A lot of parameters, and of course the tissue type, white matter, gray matter, tumor tissue, a lot of variables. So it's good that we have a lot of data. I think we need a good sense of what exactly are we after, and we need the machine learning experts to tell us how to get there.
Okay, great. Let's see.
I still want to ask Ryan that question.
Go ahead.
Ryan, do you think one day ultrasound guidance can replace MR guidance for brain applications?
It's a big question. Thanks, Song. I think I'm very excited about sort of the ultrasound guided work that's been going on. There's multiple groups now that are doing patient treatments using ultrasound guided systems, and a lot of people doing preclinical work as well. I think in the context of something like blood-brain barrier opening, where you're not necessarily getting a ton of useful information from the MRI during the procedure, you can imagine where you get preoperative images to do the targeting, and maybe you evaluate post-treatment, but maybe during the treatment it could be done outside of the MRI suite. And for something like that, of course, monitoring the bubble emissions and mapping them spatially in the brain will be very important. And that's tricky in the brain. For example, we know that we heard a lot about transcranial focusing on transmit at this meeting, but of course the same problem applies on receive. So when you're listening to emissions outside of the skull, you need to take the same considerations into account. You need to Kostas's point about capturing data. A lot of the studies that have looked to in the literature so far, at least in the brain, have for various hardware reasons, haven't been able to capture all the acoustic emissions. And we've done some early work to show that maybe that can reduce your predictive capability of the bioeffects. But I think it's also a very open question, even if you are able to generate sort of spatial information of the bubble activity within the brain, say, what signatures from those signals actually correlate to what bioeffects you're getting, whether it be BBB opening or T2 star weighted changes. And I think there's, a lot of work to be done in that area. But I'm hopeful.
All right, let's see. So I was assigned a couple of abstracts, so I wanted to point out some of them. In the abstracts I was looking at were along the lines of thermometry, and so I wanted to point out some of the work that was being done by Stephen Allen and his group. So they have an iron-based coupling medium that they put in the water to reduce the signal from the water bath, and which is great because that way they can leave the circulation on and then not have ghosting across the image. His group also has done some work with simultaneous orthogonal slice imaging as well as 3D thermometry. And so this is really fun for me to see that there is still an emphasis on volumetric thermometry, because as Crit was coming up to get his award, hi Crit, I Googled it to see that he actually published on volumetric thermometry, I think in 2009, and here we are almost 15 years later, and we're still saying this is really important, and still waiting to actually see it clinically. So I'm looking forward to seeing that. Let's see. Does anybody else have something else that they wanted to?
Wait, because the audience has any questions.
Oh, wait, somebody has a question?
No. Or
Oh, you're asking if anybody has a question? Yeah. Oh, yeah. If anybody has a question, just step right up, or a statement, something you saw that's really exciting. Let's see. Not seeing anyone. I'm going to point out something else that's really exciting in the field, is just to kind of, again, come full circle, is the idea of machine learning. And something that I've been seeing a lot is the software package that allows you to simulate across the skull with the K-wave, that they also have a part of that they're developing that's going to be real-time machine learning simulation, so that you can really just take your transducer, and as you move it around the head, it's going to do that simulation sort of in real time. And so that's just really exciting to think about, like neuromodulation studies you could do outside the MR as well. So just in terms of things that are going on in the field. Okay, let's see. Tanya, Jen, anyone want to come up with something else going on in the field right now?
May I put Hong on the spot again? Towards my question about other areas of the body that could benefit perhaps from perivascular transport. Any ideas or insights into that?
yeah, that's a very intriguing question. I haven't thought about it. So what guides us into looking into this perivascular space in the brain is because intranasal delivered agent can go directly from nose to the brain, and once they reach the brain, they don't directly go to the blood circulation. They are actually in the perivascular space. And then while we are trying to figure out how that works, then we bump into this whole field of lymphatics. So lymphatic system is currently being proposed to be the system how the brain clears its waste. So it gave us this opportunity to use ultrasound with microbubble to manipulate the lymphatic system and understand how it functions. But your question is very interesting. I would definitely look into other parts of the body, whether this can be something that we can also look into. Thank you for the question.
Okay, let's see. Cyril, do you have anything else that you'd like to add about?
I will jump in.
Go ahead, go
Oh.
No, go ahead.
I just wanted to mention pediatric care, because when I thought about what I'm really excited about that has happened recently, it really stood out to me. I don't know if it's just me personally, but I found that the midline glioma work that is coming out of Lisa Conofago's group at Columbia, and the DIPG sonodynamic therapy that we saw coming out of Dr. Packer's group at Children's National. I thought this is, treating really young children. First, it's like at the forefront of what we do with focused ultrasound, at least in my opinion, and also I think it's exciting because it has a chance of extending the capability that focused ultrasound has to impact also pediatric care, and I find that exciting. So no questions here, just a statement about it.
Could I ask a question here too? So we have this a lot of discussion here just now talking about and then Hong asked a question, taking the treatment outside the MRI scanner, right? Like, and there's a lot of research going on, right? Especially coupling with all this acoustic feedback. I do want to ask Rafi, and kind of from industry perspective, right? There's a NeuroFuss. I know that they have the neuronavigation guided focused ultrasound system, but of course the MRI guided focused ultrasound is still the mainstream. But what from industry point of view, what would it take for this, from commercialization, I guess, point of view, to take the focus ultrasound out of the MRI scanner? What do you think it would take to actually get to that and use in the clinical setting and the commercialization? And now I'm hoping Rafi and someone, maybe from the industry side can answer that question.
I didn't quite catch the question. Could you just repeat the question? Yeah, go ahead.
Oh, I'm just, yeah. Oh, I'm asking that to take the treatment outside the MRI scanner, for the brain treatment to outside the MRI scanner, right? What kind of data and also technology readiness do you think it would take for that to happen, for commercialization?
So you're hoping somebody from industry would answer this?
I'm asking Rafi, from
Rafi has
inside
something to
tech point
say.
of view. Yeah, that's what I'm asking him. I'm pointing directly to him.
Thanks for the question. So, yes, I mean, first I would say that all of us appreciate the difficulty of patients to stay in the MRI and stay in MRI for a long time. I think the answer to your question is very much related to what I raised before, which is how do we know that we deliver the treatment to a patient? How do we know that we achieved what we were set up to do, be it drug delivery, liquid biopsy, what have you? And then the MRI, I believe, still may play a role in that question. The acoustics is something that we are investing in, but we're also looking at anything else that we can put our hands on in order to be able to confirm the procedure, right? So in that sense, if I can do all of that and have a high fidelity procedure to patients that brings the patients the optimal benefit, then I have no problem leaving the MRI.
Cyril, did you have something you wanted to add?
Yeah, one of the questions we're supposed to respond is what notable application-related breakthrough have occurred recently. So we have been talking about brain stuff for 3 days now. So I really wanted to highlight a study performed in UK by the team of the Professor Lees, which is, I think, spectacular. They did not publish yet, but they are currently running a clinical trial where they want to perform hemostasis in the placenta of women pregnant with twins in order to separate the blood supply to each baby so they can grow correctly. So it's just the safety they are trying to evaluate in this early trial, but they have very good evidence of clinical efficacy. So that's, of course, it's not a huge market. There are not that many difficult twin pregnancy. But if this trial ended up demonstrating that IFU is working good, it's going to be, I think, a great highlight for our domain, for our field, with a fantastic application.
Mm-hmm. Yeah, one of the other questions that was posed to us is sort of what role do we see sort of veterinary medicine playing in terms of helping and translate these technologies, or are we just thinking about it wrong? We shouldn't even be thinking about translation, but just be thinking about veterinary medicine. What do you think, Eli?
Yeah, so we've had a lot of experience recently working with our vet hospital on multiple clinical trials, and I think, some of the advantages are the more relevant clinical data that we can get as part of that. I also think there's a lot of similarities between the pediatric cancers and the veterinary tumors that we can take advantage of as we move into both from a device development standpoint as well as just testing out diseases that move instead of relying on all of our tumor models being in particularly mice models, but some rat models, and is that going to be translatable? And we've seen that with other technologies, and I'm hoping that can be the same with focus ultrasound. It looks like we have a couple.
Okay, yeah, we have questions. Go ahead.
Hi, my name is Said, and I'm from Georgia Tech, and I would like to thank for highlighting our work, and we work on the CMAT system. And I just want to have one comment on why it took a long time to have CMATs on the applications. Actually, having or fabricating CMATs in only dedicated to CMAT is really challenging. And right now we have that actually opportunity having the clean room facility just for CMATs or the same applications. My question is different than CMATs, and I was wondering if still are we trying to solve the skull heating problem, or is there any novel methods you have in your radar right now? Yeah, and yeah, because recently there are new probes or transcranial focus ultrasound system. I was wondering if you have any new methods or any approach you are trying to solve a skull problem, especially in thermal ablation.
So you're asking if there's any breakthroughs?
Yes,
on skull
breakthrough.
heating?
Yeah, this is exactly, yeah.
Not that I know of. Does anybody know of any breakthroughs on skull heating?
Well, I'll just mention one thing that you brought up earlier, and actually he's standing behind you in line there, so he can answer it better afterward. But Stephen Allen, one of the big features of, and we have a small part collaborating with them on this, but they've really pioneered this new iron-based coupling media that can be used for the transcranial procedures. And I think one of the big benefits of that would be allowing continuous circulation during procedures without the MRI artifact. So I've been very excited about multiple aspects of that, but I think the ability to drive harder while minimizing skull heating will be a big feature of that approach.
Thanks.
Another option is to implant your transducer, but somebody did that already.
Go ahead, Stephen.
Okay, all right. Stephen Allen from Brigham Young University. I have a question about ultrasound, even though my specialty is MRI. So I'd like to ask the panel if you could envision all the experiments that you want to do in the next 5 or 10 years, all the things you'd like to do. Does existing transducer technology, is it already up to date enough that you could complete what you want to do? Or are there features or capabilities you wish you could make your transducers do that they can't do right now? So what would be your wish list?
Yeah, I have one, if I could just jump right in. Just very wide bandwidth, because there's things that we were thinking about for neuromodulation that if you could just have like half of a cycle, that's kind of a crazy idea, just, that if you could have just like half of a cycle, then it might allow you to have just positive pressure or just negative pressure. And that might be kind of interesting, but anyway, that would be very wide bandwidth. It's kind of crazy. All right, let's.
I could chip in. Yeah, completely agree. And then also from the cavitation monitoring standpoint, wide bandwidth, obviously, being able to capture as much of the spectrum we can and to use that for monitoring and control purposes. I guess the problem in the brain is that you also need it to be sensitive. So the trade-off there between the wide band and the sensitivity. But yeah, that would be great if we could have that.
I also have a dream that I hope in 5 years that ultrasound device can be as popular as Sonicator that every lab has. So I want a device that is so affordable, so easy to use, so robust, that every lab who want to use focus ultrasound technology directly have access to it.
All right, great.
And the message to the manufacturer of transducer, a cheap version is better too. A not expensive version is good.
Okay, Eli?
One thing I've been really hoping to see develop is similar to ultrasound imaging, moving to these really portable devices, particularly for some of the work we'd like to do developing for low and middle income countries or resource-limited countries having battery-powered devices that can apply these procedures in a really compact, portable setting. So I'd love to see that.
Yeah. So I have to say, just thinking about the MRI field, that there's just really a focus on low field, portability, low cost, and so it's maybe something that we should make that pivot as well, thinking about that sometime soon. Yeah. All right, let's go to Tanya.
Yeah, I would second the low cost and affordability. My biggest ask would be not necessarily the transducers themselves, but rather the electronics that would be transmit receive capable and yet also capable of a wide range of pressures, anywhere from BBB opening to histotripsy, and broadband in order to do some kind of a 3D imaging.
Okay, great, thank you.
Still affordable.
And still affordable, yeah. Jen?
Well, I echo Tanya. I think that's, there are multiple people have talking to me wonder that all-in-one system, right? Like do everything. Portable, small but high power, but transmit and receive capable, low cost, wideband. I think separately those things because we are lab investigating a lot of fabrication, design and things. Separately, I think we are able to achieve them. So came out, we actually build transducer that can do half of a cycle, positive or negative, that kind of thing. So we can talk separately later. But to do everything together, still portable and low cost, in one device, that would be great. And I think is something, I still think it's possible. And we are actually still working on it.
Great. So anything else? Any last moment comments, burning questions? Rafi, I know you always have one more thing to say.
I will comment on the last part of the discussion and disregard the cost. I think in general, we have more to do on the receive side rather than the transmit side. But in the transmit side, histotripsy is going to push us. So there are some developments to be done there. And I agree that on the receive side, we need to understand what do we do so that, especially in neuro applications, that we deal with the filtering of the skull and obtain enough signals so that if the acoustics can guide us through the treatment to well enough procedure, then we are there to pick up this signal, which is challenging.
Okay, I was just about to conclude, but I see there's one more question or a statement.
So 5 to 10 years from now, you'd like to have this distributed among all the different medical specialties and not really confined to sort of the neuroradiologists and neurosurgeons or a specific surgical group. So I think you want to make this more applicable, you're going to have to distribute it along the whole medical field to see whether or not they will want to buy into it. Number two, I think it's more important we get a handheld device. I mean, if you want to think about it from the point of view of Star Trek, you want to be able to come up and do something quickly and with a handheld type of device. But more importantly, I think 5 to 10 years from now, if you don't have the neurologist buying into this or the neuro-oncologist buying into this, this field is going to be passed by the pharmaceutical industry. And same thing for the medical oncologist, or if I just stay in the oncology field, which I am, essentially you're going to be bypassed by a variety of new types of drugs and new types of nanoparticles, and they're going to say, so what?
Mm-hmm. Right, okay, so I want to thank everybody for your attention. I also really want to put a thank you out there for all the trainees who have given all the presentations that we've gone and searched out, and thank you for all the good quality work that you're doing. And this is a very fun, exciting, and fast-moving field, and thank you for staying in it. So, all right, we're going to conclude there.